Welcome to the Explainer. What if a biological process that usually takes Mother Nature a decade could be done in just a single year? Have you ever wondered how agricultural science manages to rapidly develop crops that can suddenly withstand totally new diseases, harsh climates, and rapidly changing environments? Well, the answer isn't just traditional farming. It's actually a high-tech, microscopic solution that completely bypasses the slow, agonizing pace of traditional crop breeding. Today, we're diving deep into the fascinating world of haploid cultivation, which acts as a literal biological cheat code to rapidly develop resilient supercrops. Here is the roadmap for our scientific journey today. 1. The biological cheat code. 2. Two parallel cultivation pathways. 3. The Anther culture protocol. 4. The pollen culture protocol. 5. From cell to supercrop. And finally, 6. Impacts and operational constraints. Section 1. The biological cheat code, establishing the baseline. At the core of this whole process are microspores. Now, these are essentially just immature pollen grains. The fundamental idea here is taking a single, incredibly tiny, immature cell directly from gametophytic tissue, meaning, of course, it only contains half the plant's normal genetic material, and culturing it so it eventually transforms into a highly stable crop. So where do we actually find them? Well, if you trace a flower's long green stalk, the filament, all the way up to the very top, you'll find the anther. And if you look inside a cross-section of that anther, there are 4 distinct chambers. Those are the pollen sacs, and nestled right inside them are our golden tickets, the microspores. Section 2. Two parallel cultivation pathways, 2 roads leading to the same destination. So, there are 2 primary extraction methodologies we use in the lab. First up is anther culture, which is the intact approach. This involves culturing the entire anther directly from sterilized flower buds. Then, on the other hand, we have pollen or microspore culture. This isolation approach directly cultures the microspores without any of that surrounding tissue. But crucially, this route requires a unique pre-treatment of the anther before extraction. The real takeaway here is that despite these totally different starting points and pretreatment needs, both of these parallel roads lead to the exact same incredibly powerful destination, successfully producing haploid plantlets directly from gametophytic tissue. Section 3. The Anther culture protocol, the intact approach. Okay, so step 1 is basic extraction, but step 2, that's a strict quality control check called the acetocarmine test. You extract a sample anther, crush it in an acetocarmine solution, which actually dyes those microscopic chromosomes a deep, highly visible red, and view it under a microscope. This definitively verifies the pollen's developmental stage. If it passes, we proceed. If it fails, well, the culture is scrapped. It turns out how you plate these anthers matters immensely. And the variables shift wildly depending on your media. Solid media? That requires carefully arranging 10 to 20 anthers horizontally per dish. But liquid media jumps up to 50 anthers per 10 milliliters. On top of that, timing is totally unforgiving. Cereal species, for instance, have to be harvested precisely at the uninucleate stage. That's that incredibly brief window when the cell contains just a single nucleus. Section 4. The pollen culture protocol, the isolation approach. Now, because we physically press the anthers with a glass rod to squeeze out the microspores entirely, intensive filtration is required to remove debris. And honestly, the microscopic precision needed here is staggering. Tobacco requires a 40-micron pore size mesh. To put that in perspective, that's roughly half the width of a single human hair. Meanwhile, maize needs a 100-micron mesh. That tiny difference in screen size literally dictates the success of the entire culture. Right after filtration, we enter a strict purification cycle. The pollen suspension gets spun in a centrifuge at exactly 500 to 800 RPM for exactly 5 minutes. Think of it like an incredibly gentle, microscopic washing machine cycle. This specific low-speed spin separates a distinct, highly concentrated little yellow pollen pellet right at the very bottom of the tube. Once we've got that yellow pellet isolated, the final steps are highly streamlined. Extract, wash, and culture at a strict plating density. Finally, the whole dish is completely sealed with parafilm. This airtight seal is absolutely vital, guys. Without it, our tiny, highly vulnerable cells would just rapidly dehydrate and die. No way around it. Section 5. From cell to supercrop, biological milestones. Now we get to watch the biology take over. First, cellular division forms a disorganized lump called callus tissue. Then comes step 2. Organogenesis. This is that magical moment when actual defined organs, like tiny shoots and leaves, suddenly sprout right out of that unorganized lump. In step 3, regeneration, those tiny shoots are moved to a rooting medium. And finally, step 4, artificial chromosome doubling. Step 4 is the true biological cheat code. Artificial chromosome doubling creates a completely homozygous diploid plant. In plain English, it means the plant has 2 perfectly identical sets of genes with absolutely 0 hidden recessive traits. It's literally like copy-pasting a perfect genetic blueprint. We are condensing years of wildly unpredictable traditional inbreeding into one incredibly fast, highly efficient shortcut. Section 6. Impacts and operational constraints. Taking a step back for the macro view. So, what does this all mean? Well, the core value here is rapid trait selection. Imagine a new fungal blight is wiping out global wheat supplies. Using this method, we can rapidly lock in crucial survival traits almost immediately. We're talking intense disease resistance, yield optimization, extreme cold resistance, and precise maturity timing. We can release these engineered strains directly to commercial farmers or use them as perfectly stable parents in future breeding programs. But it isn't magic. It's rigorous science. We have to transparently lay out the 5 major operational roadblocks here. Economically, the financial costs are just massive. The infrastructure demands heavily equipped labs and climate-controlled greenhouses, all operated by personnel with highly specialized expertise. And even then, the yield of viable plants can be consistently low. Plus, there's always a lingering risk of deleterious genetic mutations occurring during this artificial process. Yet, despite those steep constraints, the overarching cycle is an undeniable marvel. By leveraging these meticulous pathways, we transform microscopic gametophytic tissue, achieve accelerated homozygosity in a fraction of the normal time, and enable rapid trait selection. It's basically an endless, accelerated loop of biological improvement. Which brings us to a really fascinating conclusion. When you weigh these incredible scientific leaps against the stark realities of laboratory economics, you have to ask, could bypassing generations of conventional breeding be the ultimate key to our agricultural survival? I'll leave you to marvel at how isolating these invisible, microscopic cells might just be our very best tool for securing the future of global food. Thanks for joining me on this explainer.